go over chromosomal changes, okay? So the first thing I want to point out that all of these are going to be different types of mutations. So that was what my last video was over, was mutations. But in that one, we were looking at, just like a mutation that's a point mutation or a simple insertion deletion of one nitrogen base being swapped out, or one being inserted, or one switching altogether. So for these chromosomal changes, we're looking at big picture chromosome changes. So like an entire section of chromosome is missing, the chromosome altogether, okay? But these are just more like bigger structural problems that are different than one base pair being different. Okay, so that's where we're going to start. But they are all different types of mutations, okay? So these are still mutations. It's a change in the DNA, and that is going to be permanent in that cell and in every cell from that cell divides to become. So that entire cell lineage will have that mutation as well. The key thing is that it is a change in the structure of the chromosome. So I have drawn pictures. None of them are very good, but you will see what I mean. So they can create challenges in reproduction. They can change a phenotype, which is the characteristic seen. So I don't know if you remember that specifically, but a phenotype is the trait that you see, like hair color, eye color, whatever. There are lots of different things that are going to be a phenotype, but it's the actual trait, the actual characteristic. Or it can have no effect on an organism. So there are going to be a couple of these that might not have an effect, and those are going to be balanced. So if they are balanced, that means that they have no effect. And we'll get to what that means later on. That's just some nice little foreshadowing for you. Now, remember that they are all going to be caused by mutagens, which are things like radiation, ultraviolet or UV rays, X-rays, chemicals, or just, classic cell mistakes. Okay, so it doesn't necessarily have to be one of these things, but it could be. The first chromosomal change that we're going to talk about is a deletion, where it is the loss of one or more genes / part of a chromosome. It's going to break off and it does not reattach. So in this lovely drawing that I made, the black part of the chromosome is original, the red part is original, but in the second chromosome that I drew, okay, it's the same chromosome, so we can say this is chromosome number, whatever. I'm going to say 7. My pen is really lagging on this video, but, so in chromosome 7, and then in the second one, you'll see this little deleted section. So that's why the red part is a lot shorter. That's why I drew it in 2 separate colors. Okay, we can pretend that the part where it meets is the centromere, but I got really lazy with these drawings. You'll see I only draw the centromere where it's needed. But that's where it happened, whatever. So part of it is deleted. It does not have to be on the end. So it might look like it's on the end, but if this is my chromosome here, right? Maybe this is the part that gets deleted, and that will result in the chromosome being shorter because it's missing that whole section. Now, an insertion is going to be the opposite. So part of the chromosome breaks off, but it is then inserted into a different chromosome. So in this drawing, I have chromosome 1 and chromosome 5. They're 2 different colors to keep them straight, and then the part that's going to be inserted, I drew in red. And then you see after the change, it was inserted into this section. So now we have chromosome 5 has the information that was on chromosome 1. An inversion is going to be part of a chromosome that breaks off on or near the centromere. So that's why I drew the centromere. And it's going to reattach backwards on the same chromosome. So that's why you can hopefully tell that that is an arrow. It's pointing up in the first chromosome. And then after the inversion, it's pointing down. So, it's inversion, it's flipped upside down. So there's going to be 2 different kinds of them. There's going to be peri with an E or para with an A. So peri is going to be at the centromere. Para is going to be anywhere else. And so this is how you're going to remember it. So para is anywhere else. That's my lovely attempt at making that make sense. But para has an A in it and it's anywhere else. So inversion, part of it's going to break off and flip. Now, translocation is going to be part of a chromosome breaks off and attaches to another chromosome. Sometimes it's going to swap places, so that's why you see these two swapped places. So we have chromosome 3 now has the bottom part of 7, and chromosome 7 has the bottom part of 3. Once again, this does not have to necessarily be the bottom part of a chromosome. It's just easier for me to draw it that way for these demonstrations, and my drawings are bad anyway, so why would I make them more complicated? It can happen anywhere on the chromosome. They don't even have to be, the same spot. Maybe the bottom of one chromosome, the top of another. Okay, as long as it is considered to be translocation where they swap or where one goes to the other, it's going to be translocation. It does not matter where it is. Now, balanced versus unbalanced. So balanced is going to be all the genetic material there is there still. So there might not be a change. So all the genetic information is still there, and that's fine. So it might have been an inversion, it might have been swapped to another chromosome, but it's still there. And sometimes it doesn't matter where the genetic information is. If it's still there, the cell's going to still transcribe and translate that information. And if that's the case, then it's all going to be fine because the cell will still produce those proteins. Now, maybe it gets put into a spot on the chromosome that it can't transcribe it. And, that could happen for various reasons. Maybe it's wrapped around histones too tight. Maybe it's close to a regulation zone that's going to inhibit it, whatever. But if it's there and it can still be transcribed, you might not run into any problem. Now, if it's unbalanced, it means that there's extra or missing genetic information. Now, that could also, and probably will, definitely cause problems, especially if you're missing genetic information. So, maybe you're missing the gene that codes for, I don't know, your skin to develop. I'm just making something up on the spot because I didn't really think about this beforehand, but, whatever. So if you're missing that genetic information, it's not going to develop. Now, if you have extra genetic information, you might produce too much of something, or, it might overtake the other proteins that, all the resources are going to this instead of something else. So that can also cause a problem. That's why there are abnormalities like Down syndrome where you have an extra chromosome and you might think on the outside, well, I don't know why that causes a problem because all the genetic information is there. But because it's extra, it's not supposed to be there, you do run into these problems. Now, the last one that I'm going to talk about are non-disjunctions. They are going to be where the homologous chromosomes do not separate properly. So this is going to happen in meiosis. Remember, we have our homologous chromosomes here. And here we have two sister chromatids. Now, if they do not separate properly here, right, you'll end up with all kinds of problems because at the first round of meiosis, if they don't separate, here's the first round, both of them are over here and none of them are here. There's nothing because none of them went there. If you have non-disjunction here, you get one that looks like this, one that's empty, one that has the proper number of and then, two that have the proper number of chromosomes, okay? That's non-disjunction. Now, I know I went through these kind of fast, but the key things to remember, these are all going to be different types of mutations. If you just, spend some time to figure out what each one means, you'll get it pretty quickly. I don't think these are that difficult. Non-disjunction, they don't separate. Translocation, they're being translated to another spot. Balanced, it's all there. Unbalanced, it's not. We've got our inversion, it's flipped upside down. Insertion, it's inserted. It's just like what it would be for the previous lecture where I went over an inserted nitrogen base. Now it's just an inserted whole string or strand of DNA is inserted into another place. Deletion, where one is deleted and it's gone, same as before, only it's once again a whole section of a chromosome. Other than that, I don't think that there's too much to cover in this section, and just keep working on our stuff.